Dear friends and colleagues, it's a real pleasure to be able to speak to you about the new classification system, the 2017 World Workshop system. And my remit is to cover periodontal health and gingivitis. This was the subject of Group 1, and you see Group 1 here. My co-chair was Brian Mealy, and it was a tremendous privilege to work with such luminaries in the field of periodontology. We set ourselves a number of tasks, but there were three main tasks. The first one was to, for the first time, define periodontal health and gingival health. The second task was to see if there was some way that we could develop a biologically based classification system rather than a traditional one based on clinical descriptors. And then the third challenge really was to try and simplify or reduce the classification from the 1999 system so that it was more easily implementable within a general practice setting. So the biology is fairly well known now. There's a lot we don't understand, but this wonderful cartoon from Steve Offenbacher enables us to visualize health. And health we now understand is associated with a health promoting biofilm. But if that biofilm is allowed to accumulate and it's undisrupted or undisturbed for a period of time, then it starts to irritate the sulcular epithelial cells lining the gingival crevice, and they send alarm signals into the connective tissues, and we see the development of an inflammatory response as you see here, and we call that gingivitis. And gingivitis is reversible, so if we reduce the biofilm or disrupt the biofilm, then it's possible to reverse the gingivitis. But then in individuals who are high risk for periodontitis itself, then the threshold, if you like, for stimulating a destructive immune response is quite low. And those patients start to develop what we generally regard as an exaggerated or a hyper-inflammatory response that destroys the periodontal attachment and alveolar bone that retain the teeth in the mouth. We still don't understand sufficiently the processes to be able to go to a full biological classification system for periodontitis or indeed for gingivitis. But the classification system in 2017 has been designed to allow for updates in knowledge to be included at a future time point. So it's fairly flexible in that regard. Now, the key dynamics that we understand for a classification system involve three main factors. Most importantly, the microbial dysbiosis, that is the emergence of disease-forming or pathogenic species within the biofilm. Then the response of the host's immune system, which, as I mentioned previously, tends to be exaggerated in periodontitis. And even more important actually than those two is the individual patient, because biologically every patient is different, and therefore their biological response, their immune response to the accumulation of those bacteria is also different. Their environment's different, their genetics are different, and their risk factors are different. So we needed to try to accommodate all three of these if we were going to move towards a biologically based classification system that allowed us to embrace what we now refer to as precision medicine or personalised medicine. Personalised medicine is why one individual is different to another. Now there is some evidence from different studies, I'm showing you data from one of our own studies a few years ago, that does enable us to map the biological phenotype of the development of gingivitis or gingival inflammation. And this was a study that was published in the Journal of Clinical Periodontology that looked at different biomarkers within gingival crevicular fluid and how those biomarkers changed as the plaque started to accumulate. And what we were able to show was that there were different biomarkers that would increase at different time points in the development of that gingival inflammatory lesion. So, for example, the neuropeptides like substance P would emerge within 24 hours of the plaque starting to accumulate, whereas some of the other biomarkers, such as the acute phase reactant alpha-1 antitrypsin, would take 2 to 3 weeks before they started to increase. But that type of biological phenotype hadn't really been developed for periodontitis at the time. And on the back of this work, we produced a hypothetical model, and that hypothetical model took us on a transition, as you see here, from health to gingivitis. And it enabled us to map the biological changes to the clinical changes we see in the mouth. So as plaque starts to accumulate and we get an initial response, that initial response is, if you like, a neurological response, and we have neuropeptide release, in this case substance P. As that plaque continues to accumulate, then we start to see the activation of epithelial and neutrophil responses, and the release of cytokines such as interleukin-1 beta, and also other biomarkers such as cathepsin G and elastase. And then as we advance towards the full 21-day of experimental gingivitis, then the acute phase reactant alpha-1 antitrypsin started to spike. So this was actually quite a clear pattern, but unfortunately we started to realise that people in practice don't measure gingival crevicular fluid. It isn't really practical to do that in day-to-day practice. And so perhaps the biological phenotype itself needed to be much simplified in the future if it was going to be used in day-to-day practice. Similarly, for periodontitis, this particular study, this particular paper was very influential in the new classification system. And this was from the Columbia group, looking at biopsy samples of gingival tissues during periodontitis, what we used to call chronic periodontitis, and aggressive periodontitis. And looking at the gene expression signatures within those mixed biopsies to see if they mapped to the type of clinical presentation of disease that we saw with aggressive periodontitis versus chronic periodontitis. And as you can see in this slide here, what the group did was they did a type of statistical cluster analysis, and they were able to identify two distinct clusters from the transcriptome work. But interestingly, those two clusters did not map across to the clinical descriptors of aggressive or chronic periodontitis, and nor did they particularly map that well to whether the periodontitis was localized or generalized. And so that was one of the factors that was taken into consideration in the periodontitis classification when it came to the decision to remove aggressive periodontitis as a distinct disease entity, because there was no real evidence that that could be justified biologically. So the current model that I like to talk about today is one in which we accommodate the accumulation of bacteria at the gingival margin and the host response. And this is a modification of the classic model described by Roy Page and Ken Corman back in 1997. And this current version basically essentially demonstrates that if you have a low biomass, a low biofilm accumulation, then the immune response to that is proportionate. It's natural, and we end up with a symbiotic relationship between the biofilm and the immune response. And so that inflammation ebbs and flows, and by and large it will resolve. One of the things we've never accounted for in a classification system, though, really is the patient's risk status, and that was very important in this classification system, because risk factors vary, and according to risk status, that can impact upon the development of gingivitis and ultimately the development of periodontitis. And so in this biological model in gingivitis, we have a higher biomass. The biofilm's been allowed to accumulate. The host response is still balanced and proportionate, but we start to see the emergence of pathogenic bacteria. So we've called this an incipient dysbiosis. And as a result of that, the inflammation starts to become chronic and non-resolving in nature. And then the final stage, again according to risk status, is the development of periodontitis. And here you'll see we have a high biomass. We have, in this case, a dysregulated inflammatory and immune response to that. So it's disproportionate, the host response. The inflammation fails to resolve because the host response starts to create damage to our own tissues. It's estimated that about 80% of the tissue damage that we see in periodontitis is host mediated. And so the various reactive oxygen species, cytokines and various other proteolytic enzymes, matrix metalloproteases, etc., are responsible for the majority of the connective tissue and bone destruction that we see in periodontitis. So based on that working model, we rapidly came to the conclusion in the first half day of the workshop that we, at least for this moment in time, needed to recognise the biology but stick to clinical descriptors for the current classification system, which is what we did.